The cell cycle is the life cycle of the cell. Most of the cell's life is spent in the phase called interphase. What is mitosis? A sexual reproduction, cell division of the cell, where one copy of DNA is distributed into each of the two daughter cells. Daughter cells are identical to the parent cell. The purpose of mitosis is to repair or replace body cells. Mitosis only occurs in eukaryotic cells. What are the stages of mitosis? Interphase, prophase, prometaphase, metaphase, anaphase, telophase, and G1. What happens during interphase? The cell grows, replicates its DNA, and prepares to begin mitosis. What happens during prophase? The cells grow, replicates its DNA, and prepares for mitosis. The prometaphase, chromosomes finish condensing so they are very compact. Chromosomes migrate to the equatorial plane. During metaphase, the cells align at the middle and it attaches to the chromatin pair of the centromere to line them up across the middle of the cell. During anaphase, the spindle fibers split apart the sister chromatids and move them to opposite ends of the cell, equally divided the genetic material. During telophase, a nucleus forms in each side and a new nuclear membrane forms around each set of the chromosomes, each with the same number of chromosomes as the original cell. Cytokinesis finishes the process by splitting the cell into two, which is the next phase, that is G1. In G1, the division of the cell cycle begins again with interphase. And what happens during cytokinesis? The cytoplasm divides, distributing the organelles into two new cells.
During the mitotic phase, the cell divides its copied DNA and cytoplasm to make two new cells. M phase involves two distinct division-related processes, mitosis and cytokinesis. Meiosis is only for sexual reproduction and stages of gamete cell reproduction. The stages of meiosis is interphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. What happens in interphase in meiosis is the cell grows, replicates its DNA, and prepares to begin mitosis or meiosis. In prophase 1, chromosomes condense, sister chromatids pair up, crossing over occurs, new combos of alleles are formed, spindle fibers form at opposite ends of the cell, and the nuclear membrane disappears. In metaphase 1, homologous chromosomes line up at the center of the cell. Spindle fibers from one end of the cell attach to the homologous chromosomes, and spindle fibers from the other end attach to the chromatid pairs. During anaphase 1, homologous chromosomes separate and move apart to opposite sides of the cell. The sister chromatids remain attached at their centromeres. This is different than in mitosis. In telophase 1, each side has set a haploid set of chromosomes. Each chromosome still has two chromatids. Nuclear membrane forms around the two daughter cells. The cytokinesis in meiosis, the cell splits into two daughter cells with two chromatids in each. The chromosomes are different from each other from the parent diploid cell because of the crossing over that occurred in prophase 1. In prophase 2, nuclear membrane breaks down, new spindle fibers form. In metaphase 2, chromosomes line up at the middle of the cell, spindle fibers attach to centromeres. In anaphase 2, centromeres separate and the spindle fibers contract and pull sister chromatids apart to opposite ends of the cell. In telophase 2, a nuclear membrane forms around each haploid set of chromosomes.